So the next concept in the chapter sexual reproduction in flowering plants after the outbreeding devices is pollen-pistil interaction. So here after understanding the process of pollination and after understanding the various interactions, various different types of interactions during the pollination, in order to have a complete knowledge of pollination, we also have to understand the process of pollen-pistil interaction, which is a very interesting mechanism that is seen in the plant body. So usually in the angiosperms, when the pollen grain falls on the stigma, so it is not necessary that each pollen grain falling on the stigma need not pollinate or need not be producing the pollen tube, need not germinate and cause pollination. So when this is the case, then you can ask a question like, so which pollen grain will actually produce the pollen tube, which will not produce the pollen tube? So this is not decided by the human beings, or this is not decided by any other factor, but this is decided by the chemical factors produced by the plant itself. And this kind of interaction wherein the pollen tube has to be produced by the pollen grain or not to be produced. So this is what is going to be decided by the interaction called pollen-pistil interaction. So you can look at the definition of the pollen-pistil interaction. It is the ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of a continuous dialogue between the pollen grain and the pistil. So that means whenever a pollen grain falls on the stigma, it need not necessarily produce the pollen tube. If it is producing the pollen tube, so there occurs a continuous dialogue of chemicals. The dialogue happens between the pollen grain and the stigma, that is mediated by the chemicals produced by the pollen grain and the stigma of the pistil. And if this chemical dialogue, if this interaction of the pollen grain and the pistil, only if it is successful, then only in that case the pollen tube will be produced. So again, so this in order to be the chemical reaction to be successful, there is again a genetic makeup that is responsible behind it. So in some plants we have seen, some plants they will undergo only the self-pollination, some plants can only undergo cross-pollination. So those plants which undergo self-pollination will support the pollination by the same pollen grain, the pollen grain of the same flower. When the pollen grains fall on the stigma of the same flower, so they will be allowed to pollinate, they will be allowed to germinate in the case of self-pollinated flowers. That means there is no sexual incompatibility. So that means self-incompatibility, it is not present there, so they are compatible by self. So that is the case. That means if the pollen grain, so just imagine, so if this is the pollen grain of plant A or flower A falling on the stigma, that is flower A. So if this is the self-pollinated plant, if it is a self-pollinated plant, in that case, the pollen grain A will be allowed to germinate and produce the pollen tube on the stigma of that particular flower A. In case, if this is a self-pollinated plant, but in the case of self-pollinated plant, the stigma B is there and pollen grain A is falling on the stigma, and pollen grain A cannot be accepted by the stigma B, and there results rejection during the continuous dialogue of the chemicals of the pollen grain and the stigma. So this A pollen grain cannot be recognized, cannot be accepted by the stigma B. So this is followed by the rejection, as I told. And in case, if it is a cross-pollinated flower, in the case of cross-pollinated flower, what would happen? So here, if this is the stigma A, and in case if it is a cross-pollinated flower, the pollen grain A, when it falls on the stigma, so this pollen grain A cannot be accepted because this is a cross-pollinated flower. This is not encouraging the self-pollination. This is going to mainly encourage the cross-pollination. So it may not be always necessarily that if a cross-pollinated plant, if it is there, it may not be always rejecting the pollen grain of the same flower. But in most of the cases, in order to encourage the cross-pollination and discourage the self-pollination, so the pollen grain of the same flower will be rejected during this pollen-pistil interaction. I hope you have understood. So this is the concept of pollen-pistil interaction. So in this pollen-pistil interaction, so we have seen that there is a possibility of either the acceptance or the rejection of the pollen grain by the stigma. So in case, so of course when it is rejected, the pollen grain will not be able to produce a pollen tube. In case, if there is acceptance of the pollen grain by the stigma, then the pollen grain will be allowed to produce the pollen tube, and this pollen tube will move all across the style, and it reaches the ovary, within which ovule will be present. And so in this ovule, so it's going to enter from the micropyle of the ovule, and so once it enters into the micropyle of the ovule, it's going to cause the pollination, or fertilization is going to be caused. So you know the structure of the ovule. So the pollen tube entering into the ovary, so will enter into the ovule through the small pore called micropyle. So through this, when the pollen tube enters, it will later enter into the female gametophyte or the pollen sac, what we call it as, and then after entering into the pollen sac, so the two male gametes or two sperms which are carried by the pollen tube, so they are going to be released into the female gametophyte. So one will fuse with the egg, the other will fuse with the central nucleus. So that we will understand later in the double fertilization concept. So the other concept, the next concept in this chapter we are going to study here is artificial hybridization. So after understanding the pollen-pistil interaction, we are moving on to the next concept, that is artificial hybridization. See, this is a concept where we actually practice. So in this artificial hybridization, it is a practice of conventional plant breeding. So conventionally, so how the plants were hybridized even before the invention of advanced technique of hybridization of plants. Like today we have got tissue culture, we have got cell cultures. So various different advanced techniques we have got in the plant sciences in order to hybridize the plants, but this is one of the oldest hybridization techniques we have followed. So this mainly involves two main steps. One is called as emasculation and the other is bagging. So in the case of emasculation, what do we have to do is, so we have to select a healthy plant. We have to select a healthy plant on which we have to select a flower in the bud condition. That means it is not yet opened. The flower should be not open. And one more very important condition what we need to see here is that the plant body should be such that it should be consisting of the flowers, and these flowers should be bisexual. That means the flower should be having both the androecium and gynoecium. If you select a plant, either male plant or female plant, so you cannot practice this particular kind of artificial hybridization in those plants, and it is not necessary there also. So here, if you select a plant consisting of both androecium, gynoecium, and this flower is in the bud condition, and this bud of a flower will be opened very slowly without harming any appendage of the flower. We are not going to cut off the petal, we are not going to cut off the sepals, so we are not going to harm to any part of the flower. So slowly we will open up. So you can look at this diagram. So this is the opened flower. You can look at immature. That means because the flower is still in the bud condition, the androecium and gynoecium are not yet mature. So these immature androecium, gynoecium we are going to see once we open up the flower artificially by using the forceps. And we are going to make use of a scissor and we are going to cut out all the stamens. That means the male reproductive part or the androecium part will be removed from this flower. We are not going to disturb the female reproductive part. We are not going to disturb the petals or sepals. We are only removing all the anthers from that flower. And this step of removal of the anthers is what is called as emasculation. So we are going to remove one by one all the anthers from this flower, and this is what is called as emasculation. And after this emasculation is done, what are we going to do is, so we are going to bag it. So we are going to make use of a polythene cover or a cover made up of a butter paper, and we are going to bag it. We are going to cover that emasculated flower. So why are we doing this? See, our main aim is that we don't want this flower to get self-pollinated. See, you can look at the heading, it is artificial hybridization. Hybridization means what? So hybridization is the process in which two plants are crossed. That means the male reproductive system or the male gametes of a different plant and female gamete of another plant, they are going to be fused in order to cause the progeny, in order to bring about the progeny. So that is what we are doing. So we are doing it artificially. So we are removing all the anthers, thereby giving no chance for self-pollination, and we let this flower to mature. That means stigma has to become receptive. So until then, we are going to cover it with the polythene paper or a butter paper bag, so that there are no other pollen grains coming and falling on the stigma. We don't want any contamination by unwanted pollen grains here. So that is why we are going to bag it. And after some time, so of course, when we bag it, we are also going to put a tag. So tag consisting of some information. What information? So name of the plant, the scientific name and the common name, maybe Rosa indica or Solanum melongena, Solanum indica, whichever plant it is, and along with the common name, and name of the breeder. So who is breeding that plant? Location, timing. So timing is very important. Date and time is very important, because so you should know approximately when, after how many days the stigma is going to mature. So by looking at the date on the tag, so we are going to open up this bag and we are going to pollinate. We are going to dust the pollen grain of a desired plant. We are going to bring the pollen grain from a desired plant. We are going to dust on the stigma and we will let it to pollinate. Again, we will cover it with the same kind of polythene bag or the butter paper bag. Again, to avoid the contamination by unwanted pollen grains. So this flower will be pollinated flower. Now pollinated flower will be covered until it matures, until the pollination occurs, until the fruit is produced. So this is a kind of what artificial hybridization we will study. There are many other kinds of artificial hybridizations, so we are going to study in later classes.